former graduate of our same department. She did a biology degree in biology with a minor in GIS. So today she'll be presenting a thesis on the spatial distribution of soil carbon and its relationship with above ground carbon stock and species composition at Busby Forest. The way we go, when I give Lakeitha time to present, after she's done, probably 15, 30 minutes or more, we don't know. After she finished, then you'll be given 5 minutes. The audience will give 5 minutes to ask questions, 5-10 minutes. After you finish your question, then we ask you to leave for a while, and then the committee will interview the student. After the interview, and we're gonna ask the Lakeitha to leave for a few minutes, then we're gonna make a decision. She's gonna come back. After she can come back, after she comes back to find out whether the committee agrees on her thesis or not. Okay? Okay, so.
Hello everyone, good morning. The title of my thesis is Spatial Distribution of Soil Carbon and its relationship with above ground carbon stocks and species composition at Busby Forest. And this is just an outline of my thesis that I'm going to present to you. Introduction, literature review, materials and methods, results, and summary and conclusion. Forests and soils are major sinks of atmospheric carbon, and thus the influence of forests in the global carbon cycle is now widely recognized. Soils contain the largest pool of terrestrial organic carbon in the biosphere, storing more carbon than contained in plants in the atmosphere combined. Soils also contain about twice the amount of carbon present in the atmosphere, which makes it an important carbon sink that plays a critical role in mitigating gases. Studies have suggested that if productivity rises in the forest ecosystem, more carbon may be removed from the atmosphere and stored in trees. Assessing carbon accumulation in trees and soil in secondary forests, such as the one at Busby Forest of Lincoln University. It may pose as an exceptional challenge since there is no previous information on the area occupied by the forest and the tree species it contains. The goal of this study was to understand how tree species affect soil carbon dynamics at Busby Forest of Lincoln University. Specific objectives are to determine the composition and distribution of tree species, assess the amount of carbon that is sequestered in these trees, also evaluate carbon stored in the soil, and understand the relationship between tree biomass and soil carbon. It was hypothesized that Busby Forest is composed of several tree species. The tree species contain less carbon than soil, and there is a strong correlation between soil carbon sequestered in trees and that in soil. Many statistical tools have been used in the quantification of spatial patterns and changes in soil organic carbon. Measuring the quantity and the spatial distribution of soil organic carbon is essential for evaluating soil and ecosystem functions and for understanding soil carbon sequestration processes. For many years, it has been recognized that the spatial pattern of soil organic carbon is controlled by environmental state factors. Carbon dioxide is increasing in the atmosphere due to human activities and trees, which can be used to reduce emissions in two ways. The first way is the direct carbon sequestering by reforestation and afforestation that yield a stock of carbon in standing trees. The second way is the use of forest products as substitutes for fossil fuels or fossil fuel intensive goods such as steel and concrete. The most accurate method for estimation of biomass is through cutting of trees and weighing of their parts. This destructive method is often used to validate others, less invasive and costly methods, such as the estimation of carbon stock using non-destructive in situ measurements and remote sensing. Busby form is intended to be the demonstration form for integrated agriculture and extension. I had the pleasure of doing my research here. The field was initiated in late August 2010 in 0.49 hectares of the Lincoln University's Busby farm. This farm is also located 10 miles from the main campus. Here is an image of Busby Forest, and this is the entrance, and this is the compost here. But where the trees are distributed is right here, and I put it according to genera. Estimation of above ground biomass is an essential aspect of studies of carbon stocks, the effects of deforestation, and carbon sequestration. Weighing tree biomass in the field is without doubt the most accurate method of estimating above ground tree biomass, but it's extremely time consuming and a destructive method, generally limited to small areas and small tree sample sizes. We estimate our above ground biomass using allometric equations. Allometric equations relate tree diameter at breast height or other easily measurable biophysical variables such as commercial boil height and wood density to standard value of wood or total biomass carbon. Recent studies have suggested that forest structure changes significantly with their environment. The diameter at breast height was used in the calculation of tree biomass in this study. The diameter at breast height is affected by environmental factors such as temperature and water, since these factors affect plant growth. Carbon stock is typically derived from above ground biomass by assuming that 50% of this biomass is made up by carbon. This particular site is a secondary forest that is dominated mainly by oak, hickory, elm, cedar, and ash. Trees with various understory, native, and invasive species commonly associated with forested conditions. Previous study also showed that this particular forest was mainly dominated by oak and hickory only. We inventoried 425 trees at Busby Farm of Lincoln University, belonging to 23 different species. This is a list of the tree species at Busby Forest. The first column indicates the common name. The second column is the scientific name. The third column is the number of trees distributed throughout Busby Forest. And as you may see, the asterisk, which indicate that these specific trees were dominant throughout Busby, which we have American elm, chickaping oak, eastern red cedar, green ash, northern red oak, shagbark hickory, slippery elm, and white ash. This is an image of shagbark hickory that I took, and as you may see where it gets its name from, the shagbarks. An image of the understory of black oak. What we did, we tagged each tree. We measured its diameter and breast height. We calculated biomass of the following tree growth parameters using allometric equations found in the North American Carbon Database. Total above ground biomass, stem biomass, foliage biomass, branch biomass, and root biomass, which is 20% of the total above ground biomass. We collected soil samples at 250 locations and measured total carbon and total nitrogen. We air dried the samples and to measure total carbon and total nitrogen. We also test seed. This is a seed that we use to seed the soil, which we ship to a commercial laboratory for analysis of total carbon. The quality assurance and quality control that this specific laboratory used was that they ran a blind quality control for every 100 samples. The instruments were tested running standards prior to determination of carbon to nitrogen ratio. So they also used the North American Proficiency Test Program, and the main purpose it was to provide double-blind check samples to laboratories which wish to monitor and improve the quality of their soil testing data. Once the analysis of total carbon was completed, values for soil carbon fractions was calculated using previous data on soil carbon available as before us. So what we did was develop the Trance surface model for soil total carbon and its fractions, using data from a previous study at the same site. This model explains soil total carbon and its fractions as functions of latitude and longitude of each sampling location. We predicted the following soil carbon fractions: hydrological carbon, non-hydrological carbon, cold water extractable carbon, hot water extractable carbon, at unsampled locations using the Trance surface model. We also developed additional equations between total carbon and its fractions. Trance surface model, it approximates points having known values with a polynomial equation. The equation can be used to estimate values at other points. A linear or a first order Trance surface uses the following equation, where the z attribute value z, is a function of x and y coordinates. The b coefficients are estimated from the known points. So I use Statistics 9.0 and we get a linear regression of total carbon. As you see, we have the predictor values, the constant latitude and longitude, and it gave us these coefficients, which we had an R square of 0.53, 50% coefficient of determination indicating variability. We also that it was significant at a less than 0.05 p-value. So this is what I did. I got these coefficients and used these equations to calculate total carbon due to the fact that we had longitude and latitude. And I did the calculations, and this is what the total carbon gave me. This was the total carbon that I got for this particular tree. We analyzed the data using standard procedures with the aid of Statistics 9.0 statistical analysis at a p-value of less than 0.05 significance level. We conducted a summary of statistics, analysis of variance, and correlation analysis between the carbon fractions, total nitrogen, tree diameter and breast height, and plant growth parameters. Okay. This is a table of the descriptive statistics and analysis of variance for diameter and breast height for all the trees, for all tree species. As you may see that dogwood, the mean for diameter and breast height range from 6.77 for dogwood all the way to 39.48 for post oak. You may see that hackberry, honey locust has the highest, but it was only one tree distributed throughout the forest. One-way analysis of variance was used to test for the tree species differences among tree diameter and breast height. And the tree species for tree diameter and breast height differ significantly at a p-value that was less than 0.05 for each species. So this is a table of the comparisons of means for tree species group for diameter and breast height. The pairs with that significantly are different are flagged with an asterisk. As you may see, that black walnut was significantly different for all trees, but not for chinkapin oak, green ash, northern red oak, and also white oak. This is a descriptive statistics and analysis of variance table for total above ground biomass. As you may see, the total above ground biomass lowest average was 10.92 for dogwood again for this particular parameter, and for post oaks 731.14 kilograms. And also for the analysis of variance, we see that the trees were significantly had significantly different means. So this is the comparison of total above ground biomass means for all tree species. For chinkapin oak, you will see that it did not. It had the same mean for green ash, northern red oak, and white oak, but for all the other trees, it was significantly different. This is a table for statistics descriptive for an analysis of variance for foliage biomass. And as you may see here, that hackberry, with the coefficient of variation, it ranged from 47.21 for black cherry to 129 for hackberry. And as you may see, that the comparison of means for the analysis of variance for the species were significantly different. Here, if you see this is the comparison mean for foliage biomass with hackberry. It was significantly different only with one specific tree, and that's with chinkapin oak. This is a statistics and analysis of variance for stem biomass. As you may see here, that the coefficient of variation ranged from 52.44 to 148.55 for both sugar maple and hackberry. And also for the analysis of variance, you see that the comparisons of means were significantly different. As for hackberry, this time for stem biomass, we see that hackberry is significantly different from black oak and chinkapin oak only. This is the statistics and analysis of variance for branch biomass. And the mean value ranges from 2.81 for dogwood and 118 for post oak. And with the analysis of variance, significantly different means. And as you see right here, that post oak, it was significantly different for all trees except black oak. Last but not least, we have a descriptive statistics and analysis of variance for root biomass. And you see a variance from 5104 to 143 143.65. For sugar maple to hackberry. And with black oak here, you see that it's significantly different for all trees except chinkapin oak, green ash, northern red oak, and white oak. This is a summary of statistics of total nitrogen and carbon fractions. We see here at the mean that total carbon was significantly had the highest average amongst the other carbon fractions, and also higher than total nitrogen. As you see the coefficient of variation, that it ranged from 5.99 to 35, so it showed tremendous variability for the total carbon and its fractions. This is a correlation of diameter of breast height and plant growth parameters, where you see the diameter of breast height was significantly correlated with all the plant biomass, the plant growth parameters. The correlation of total nitrogen and carbon fractions, you see that total carbon and total nitrogen and its fractions were all significantly correlated amongst each other. This is a correlation of total carbon, total nitrogen, diameter of breast height, and plant growth parameters. You see that total carbon was significantly correlated with only DBH and foliage biomass, but not with the other plant growth parameters. For total nitrogen, on the other hand, was significantly correlated with DBH and the other plant growth parameters, but branch biomass. This is a correlation of hydrological carbon, non-hydrological carbon, diameter of breast height, and plant growth parameters. You see that hydrological carbon and non-hydrological carbon both were significantly correlated with diameter of breast height and foliage biomass only. Correlation of cold water extractable carbon and high water extractable carbon, diameter of breast height, and plant growth parameters. You see the same trend going on here, to where that both cold water extractable carbon and high water extractable carbon were significantly correlated only with diameter of breast height and foliage biomass. This is a variogram of soil total carbon. It shows the range, which is 1.20, is which the maximum distance across the field that you can go before you will see a change in the data. So data will remain constant once you reach the 1.20. As for total nitrogen, the range is 15.9 to where you will see a change. Once you reach the 15.9, you will not see a change in data. Carbon to nitrogen ratio, range 11.0, the maximum distance across the field. Before you will see change, once you reach that, you won't see a change. For total above ground biomass, the variogram shows that is the range is 125, and that's also the maximum distance before you will see where you won't see a change. So here we have a map of diameter of breast height and total above ground biomass. With diameter of breast height, it shows that the trees were bigger along the fence of Busby Forest, and the trees seem to get smaller as you go downhill. The map for total above ground biomass shows that the biomass was higher in these areas due to the bigger of the trees. This map shows total carbon and total nitrogen, and total carbon indicates that there's less distribution of carbon in the sampling area. For total nitrogen, the map, which indicates basically that the same thing, but in the interest of, In the interest of buzz before, as you see, it's a greater amount of total nitrogen present. This is a map of carbon to nitrogen ratio, which it seems to be a little scattered because of the measurements of both carbon and nitrogen. In summary, we inventoried 425 trees. We had 12 dominant tree species amongst buzz befores. The DBH was significantly correlated to all plant biomass. All of the carbon fractions were significantly correlated with each other and also with total nitrogen. Carbon to nitrogen ratio had the lowest coefficients with total nitrogen. Total nitrogen was significantly correlated to diameter and breast height, total above ground biomass, foliage biomass, stem biomass, and root biomass. Total carbon was significantly correlated to diameter and breast height and foliage biomass. The total carbon, total carbon variogram showed the range 1.20, the maximum distance across the field that you could go before you would see a change in the data. Total nitrogen range was 15.9. The carbon to nitrogen ratio range showed that it was at 11.0. Total above ground biomass showed that the range was at 125.4. The map showing the distribution of DBA showed that the trees were bigger along the fence of the forest. Total above ground biomass map showed higher levels of biomass along the fence because the trees were a lot bigger. Total carbon map distribution showed less carbon distribution, and total nitrogen did as well. Carbon to nitrogen ratio showed major differences across the field, and this is because of measurement of both carbon and nitrogen. In conclusion, plant biomass is a controlling factor for soil total carbon, and this fraction that buzz before. Therefore, there is a direct correlation between the stock of carbon in trees and those in soil. Further studies will be conducted to improve the correlation between biomass and total carbon, and also assess the relationship between biomass and total carbon by species and carbon depth. And these are my acknowledgments. Thank you.
Okay, so do we have time for the audience to ask questions first?
Yes, Stephanie.
Can you explain to me what afforestation is?
Afforestation is basically where trees have never been there, and it's the building of new, planting of new trees.
Yes, question.
Yes.
Could you have taken your measurements at a different height besides the DBH and like obtain the same results?
No, because the thing with the North American Carbon Database, with this particular system, you have to have the tree species and you have to have the diameter and breast height in order to calculate the plant growth parameters. And say, for instance, with American Elm, when I input that into the database and also put the DBH, it didn't give me any information. But when I enter white elm, which is another name for American elm, it actually gave me the information that I needed for the biomass. Thank you.
Okay, so if there's no more question from the audience, we ask you to stay for a few minutes. You can go to my office or the other anywhere. So you can come. Ask a question. Okay, so we'll invite the. Can we close the door?